I'm going to explain how to fill in the lovely poster you've got all about carbohydrate digestion and its absorption from the small intestine into the blood. So first of all, we need to know about the breakdown of starch. That's in the syllabus. Key point. So first of all, we need to mention the enzyme that actually breaks starch down into maltose, and that is amylase. There's amylase released in the salivary glands, and there's also amylase released in the pancreas. And we've seen some past paper questions where you really need to read carefully which one they're wanting you to mention, okay? So the large starch molecules are broken down into the disaccharides maltose, and then these need to be broken down even smaller, and we need another enzyme, which is maltase, breaking down the maltose finally into monosaccharide glucose units. Okay, and all of these chemical reactions are done by enzymes, and enzymes break down molecules by hydrolysis, adding water to the system. Actually adding water to the system, that's right. Okay, we also need to know about a couple of other disaccharides and how they're broken down. So there's the disaccharide lactose that's broken down by lactase, and the two monomer units that is made up of is also glucose and also galactose. And then we finally got sucrose, which is like the sugar in your tea, and we have the enzyme sucrase. You've got to be careful. If your spelling is not great, you've really got to be careful that the examiner can clearly see the A rather than the O. So no matter how bad your spelling, you've got to make that clear, otherwise you could lose a mark. Okay? So sucrose is made up of glucose and the 5-carbon sugar fructose. Okay? And that in a nutshell is everything that you need to know from carbohydrate biochemistry. So now we've got these small glucose molecules, we need to learn, or not just glucose, small sugar molecules. How do they actually get from the gut into the blood and transported around the whole body? We need to look in detail at movement across the epithelial cell in the gut. So here we've got the lumen, so the hole, if you like, inside the small intestine. We've got these epithelial, I put here intestinal epithelial cells, and lastly down the bottom we've got the intercellular fluid or the blood. Okay, so we're going to start off with easiest things first of all. So first of all we'll talk about this movement from a high water potential to a low water potential. This is a special type of diffusion involving just water molecules, and it's called osmosis. So the type of movement is osmosis. It very much is moving down a concentration gradient from high to low. It doesn't need a special protein at all, and it's a completely passive process. There is no energy spent on this process at all. Another relatively simple movement across the membrane is this one here. We've got high oxygen concentration going to a low oxygen concentration. This is just simple diffusion. It is checking my spelling there. And we don't need a protein to help or facilitate the movement of these small molecules across at all, and the process is completely passive.
Miss, that's the wrong box.
No, it's not.
That's the box for...
Oh, you're right. Okay, thank you for spotting that. Okay, I'll just put a little arrow. That one is that one. I do apologise, ladies and gentlemen. I'm not going to start re-recording though because of that. Okay? No. Shush. Right. Okay, we'll do that more complicated box later on. I do apologise. So we should really, if I have in here This basically should have all gone down to here, shouldn't it? Okay, we'll come back to that. Hopefully it will make sense when you go through this. So let's go to another fairly okay one down here. We've got a high glucose concentration going to a low glucose concentration. You'll see there is a protein involved. It is diffusion because it's going down a concentration gradient from high to low, but it needs to be facilitated by a protein. So the type of movement is facilitated diffusion. The type of protein is a carrier protein. And the process again is completely passive. No energy is spent whatsoever. We will now look at a process that does involve energy. Okay? And this is the example here. You'll see that it's moving from a low concentration to a high concentration of sodium ions, and the potassium's moving from a low potassium concentration to a high potassium concentration. It's going against the concentration gradient. So this type of movement does require energy, and it is an active process. It's called active transport. Okay, the process is active. Energy needs to be spent. We'll talk about that in a minute. The type of protein, strictly speaking, I suppose, because it is using charged particles, we could say a protein channel, but you'd have probably got the mark quite happily if you'd just put a carrier protein as well. Okay, it does require a protein of some sort. Okay, so protein channel. So, thinking about the active transport, cells that require a lot of energy have lots and lots of mitochondria. You need to recognise this organelle because this is commonly asked, okay? So there are lots of mitochondria in epithelial cells, and they provide ATP, or adenosine triphosphate. Simply speaking, this is energy. And this energy is gained from respiration. This is the site of respiration, the mitochondria, which we'll learn about in A2. And this is required for active transport. In your revision sheet that I've given you, you'll see there are a number of cells that require lots and lots of mitochondria. So examples would be muscle cells, the ciliated cells of your airways, because the cilia are constantly moving, they need mitochondria as well for that process. So there's lots of different examples in the body of cells that need mitochondria. Okay? So let's come on to this type of diffusion going on here. You'll see it's going from a high concentration to a low concentration. So this is diffusion, but it needs a protein. So it's facilitated diffusion. You'll see they're charged particles. So the type of protein, or the carrier protein, is actually called a protein channel. But it's happily going from high to low, so it's a completely passive process. We're saying it's facilitated diffusion. I can't speak and write at the same time. Facilitated diffusion, and the type of protein is a protein channel. Okay, which brings us now on to the last type of movement, which is the trickiest one. Okay, this is the secret passage, I call it, because there's two different things going on. The active transport sodium-potassium pump here actively pumps the sodium out of the epithelial cell to make sure we have this low sodium concentration maintained in the epithelial cell. This means there's always a concentration gradient of sodium from the lumen of the gut into the epithelial cell. So sodium wants to move down this concentration gradient. And there's a very, very special type of protein called a co-transport protein that will happily take the sodium through down the concentration gradient. But when the sodium binds, it actually allows a glucose to be co-transported at the same time, even though the glucose is going against its concentration gradient. This means the body can take up all of the glucose. Remember, it's a very precious commodity. We need all of it. We do not want to excrete any glucose at all. It's precious, precious stuff. So we've got the type of movement is actually co-transport. Remember, I'm filling in the wrong box. These are around the wrong way. But the type of movement is actually co-transport. Okay, there is a protein needed. It's a co-transport protein. Some of you may have come across the term a symport. It's sometimes used in some books. And most importantly, the process is passive. Even though the glucose is going against the concentration gradient, it sneaks in. The actual process of the high sodium concentration going to the low sodium concentration is down the concentration gradient. No energy is needed to be spent. It is a passive process, but it relies on this active process down here, ensuring this low concentration down here. But the co-transport protein itself is passive. And there we go. Hopefully you found this useful and you'll find it useful in the future. I'm going to go now.